get your handy dandy atlas ready here. And we went over a bunch of this stuff in the lecture. So this is kind of like a nice little bit of review for us. All right. So what page is that on in the book? Did you say 31? 41. 41 for you at home. So we're going to start off, and you can see on the picture here we've got, all right, well, as much of as a full neuron as we could possibly have here. And then we zoom in on it, all right, on the center of the picture. So our cell body, or the soma, all right, is going to be this centralized structure inside the black circle. You can see we have a nucleus in there. We have our endoplasmic reticulum along with some Golgi apparati there. But you can see, well, it's tough. I'll show you on the next, on a slide here soon, a little bit more. So this is a nice bulging region here. And this is where the graded potentials are going to occur in the cell body. Okay, so what looks like a kind of like a clear golf ball structure there at the center of the soma or cell body, that's the nucleus. Okay, our control center where all the DNA and all the fun stuff is happening. All right, now what we're going to do is we're going to shift into the axon, this long portion here. And if you zoom in, well, if you zoom in, if I zoom in, okay, you can see this right here is the mitochondria. Okay, what's the nickname for the mitochondria? Powerhouse of the cell. And also, J.J. Watt on his Twitter feed said, mitochondria is the powerhouse of the cell, which leads me to believe that he's going to be playing for the Buffalo Bills next year. Just thought I'd throw it out there at you. It'd be nice if we could have him playing for us. All right, but he really did say the mitochondria is the powerhouse of the cell. It was very cryptic, and I was like, wow, is he taking Bio 210 also? That's pretty cool. All right, so the mitochondria is our cell's powerhouse. We go back into the cell body here, and let me zoom in here. It's kind of tough to see, but on the endoplasmic reticulum, you've got these little, now the arrow's pointing at the blue structure. It should be pointing at these purplish, reddish dots. All right, this is a free ribosome here. Okay, so make sure that when you're labeling it, when you're talking about the chromatophilic substance or the Nissl bodies, okay, in case you want to see how it's spelled, that it's not pointing to the blue thing, that it's pointing to those dots. And that's all it is. It's a ribosome. Okay? And when we use chromatofin, which is a type of dye, all right, the histologists use, all right, picks up a reddish color tint to it. Okay, so remember what we said in lecture here, the cell body has these smaller processes that kind of move off or pop off of the soma here, and those are the dendrites. Okay, and their job is to transmit, well, their job is to receive the information and then transmit that information to the cell body in the possibility of generating a graded potential. Now, I know that you folks keep hearing me say that, and you're probably really excited, and you want to know what a graded potential is, but we'll talk about that on, what's today, Tuesday, Thursday, hopefully, okay? You just have to wait. I'm sorry. Okay. So, moving on down the line here, the arrow here is pointing to this white structure. That's the axon. The axon includes all this stuff on the inside too. Okay? Think of it like a wire. That's simple. Okay? It's a wire, and it includes everything about that wire, like your cell phone's charging cord. It's the whole cord. Okay? Now, your cell phone's charging cord or a wire in the wall has a rubber, all right, covering to it. In this case, the covering to our axon, which is the actual plasma membrane of the axon, is the axolemma. All right, that's this white part. It doesn't include all this stuff on the inside, because that's the cytoplasms on the inside, all right, with these organelles in there. It's just indicating this white part here. That's the axolemma. All right, if you remember, when we go down the axon, all right, at the end, you have these little branches that split off. And on the end of those branches, you have those little knobs there, okay? So those branches, we refer to those as the telodendra. Now you're seeing, all right, they're brown here on this picture here. They're telodendra from another axon that we just don't see. Or another neuron, excuse me. Okay? But we know that the telodendra, all right, are just these extensions, these branches of the axon. And at the end, it has the synaptic knobs, which are these guys. We'll label those in a second. Okay, so that skinny-like portion here, that's the telodendra. All right, so there you can see the knob. That's swelling at the bottom. Okay, you can call it the synaptic knob or the synaptic end bulb. And that's where the secretory vesicles are stored, which are hanging out with our neurotransmitter. Okay. So this other part of the neuron, all right, where you start to see the cell body taper down into the axon, that's the Axon Hillock. What is significant about the Axon Hillock? That's right. That is where we initiate our action potential. Okay? We initiate the action potential, and then it just heads on down the axon. Our electrical signal starts here in the Axon Hillock. Okay. So in our peripheral nerves, okay, some of the neurons are covered by a cell. And the cell is called a Schwann cell or neurolemmacyte. And that Schwann cell or neurolemmacyte, either or is okay if you want to answer that, okay, on a test. But the Schwann cell neurolemmacyte creates myelin. That's that fatty sheath that covers the axon. It helps to insulate, all right, the axon to speed up, all right, the action potential. We'll talk about that in lecture 2. So a cell, like any other cell, all right, or most cells, I should say, not like any other cell, has a nucleus. And that's pointed there. The nucleus looks kind of red. It's going to be on the periphery because these cells wrap themselves around the axon. If you've ever made pigs in the blanket, kind of like that. Taking a little hot dog weenie and rolling it up in some, I don't even know what they use. Is it a croissant roll? Okay, all right. I love croissants. I just want to let you folks know that over the spring break, while you're probably studying or relaxing maybe, I am taking a bread baking class. I'm going to learn how to make croissants. Honest truth, because that's the only kind of breakfast sandwich that I like. Well, I won't bring them in if they're garbage. That's for sure. I don't want you guys judging me. All right. If they're really good, I might just bring it in. Okay. So, like I said, the Schwann cell of the neurolemmacyte creates the myelin or myelin sheath. Okay. So some of you might be concerned, like, oh my gosh, you know, the arrow. If you look, this slide here. And this slide here, the arrow didn't even move, right? It's pointing to the same darn thing. Okay, don't worry, I got your back. All right, because if the question says, name the cell, then you'll say Schwann cell or neurolemmacyte. Okay, so if it says name the cell, you'll write one of these down here, this one or this one. Okay, if it says name the covering or the contents, then you're going to say myelin sheath or myelin. Okay, cool? All right. Just, I don't want there to be any confusion. I know the arrow did not move and it's pointing to the same darn thing. And people get, that's why, again, first thing you should be doing, if you're going to take the test tonight, if you haven't taken it already, or tomorrow, the lab test, scan the slide. 2-second scan. Is there any writing on this slide? No? Okay. Straight up identification, whatever the arrow is pointing to. All right? If there's writing on the slide, read the writing. Okay, so You notice here, there's an indentation here and over here, okay? So this bracket is indicating from this indentation here to this indentation here, this whole region here. That's called the internode. All right? And I'll show you what a node is here. Okay, so this pretty much this whole area where the Schwann cell, that myelin sheath is covering, that's the internode. Okay, it's a space. Here's what a node is. You can either call it the node of Ranvier or a neurofibril node. All right, that's that indentation there that you're seeing. When I first took anatomy and they were teaching us about the physiology of what's called saltatory conduction, they taught us that the action, we'll talk about that, the action potential jumped from node to node, but it doesn't do that. So lots come in advancement of our understanding of the mechanisms, right, of nerve conduction. And then finally, our last structure that you need to know is that gray structure. We talked about that in lecture. That is one of the 3 connective tissue layers involving nervous tissue. And this is that endoneurium that is going to cover, it's made up of that areolar, which is a loose connective tissue that covers each individual neuron. Helps to insulate it, all right? And it separates each neuron physically from its neighboring neurons. Okay? That's it for the neuron. Not bad, right? Not too bad, not too bad. All right, so let's talk about some neuronal anatomy. All right. So this slide is the exact same slide that you just wrote down in lecture. So you don't really have to really write too much down on this, but I do want to emphasize again the definition of what a nerve is. A bundle of parallel axons in the central nervous system. Parallel axons in the central nervous system. Okay? So when we're dealing with nerves and ganglia, we're dealing with the peripheral nervous system. Okay? And so there's 3 coverings. We just labeled one, this guy right here, the endoneurium. Okay, that's the wrapping that covers, right, the individual axon. Okay, made up of that loose connective tissue, the areolar, and it helps to physically separate and insulate each axon from one another. Okay, so now we're going to take a bunch of axons, we're going to group them together into what we call a fascicle. Okay, it's a bundle of axons. And then we're going to wrap around that thing dense irregular connective tissue. A little stronger, a little hardier, cool. And then we're going to take all the fascicles and we're going to wrap them up nicely into what we call the epineurium, which goes around the entire nerve. And again, it's made up of dense irregular. Okay? So inside of our epineurium, okay, and the perineurium, that's where we're going to load in our capillaries. Okay, so we can get the nutrients, all right, and the substances. Because remember, we just learned about, all right, some of the substances that neurons need. Sodium, potassium, okay? Blood carries that. If you've ever seen your blood chemistry panels, check it out. Make sure that your numbers are right. All right. So, quickly, here you can see all those different layers here. Here's the epineurium. Here's our fascicles, which are covered by the perineurium. And then our individual axon coming out, which is going to be covered by the endoneurium. All right, again, you've already seen this slide here, so I'm not going to spend a lot of time on this, okay? But when we're talking about, since we're going to be focusing more so in lab on the structural, the anatomy portions, okay, when we talk about nerve classification, easy, keep it simple. Remember the KISS principle, keep it simple, okay? If it's coming off the brain, it's cranial nerve. If it's coming off the spinal cord, it's spinal nerve, okay? All spinal nerves are mixed nerves. Okay, what's a mixed nerve? All right, it has both sensory and motor neurons. Not all cranial nerves are mixed nerves, though. Okay? Cranial nerves can either be sensory, motor, or mixed. If we have time, not today, but we'll talk about that. Okay? So keep in mind, sensory nerves have only sensory neurons. Motor nerves have only motor neurons. Individual axons have to be one type. It's either motor or it's sensory. It cannot be both. Okay? All right, and then our definition of a ganglion. Okay, again, cluster of neuron cell bodies. Now we know what a cell body is, right? Because we just saw the anatomy. We get that now. Okay, cell body. All right, what comes off the cell body? The dendrites. Okay, so keep in mind, this just leads me to remind myself, all right, that myelin, which we just saw, the Schwann cell, right, myelin will not be found on cell bodies or dendrites. Those areas are always unmyelinated. So that means a ganglion is always going to be unmyelinated. So it's going to appear to have, on gross inspection, right, on gross inspection, it's going to appear to be gray matter. Okay? It won't be white. White matter is only myelin-covered structures. All right, good. This is a nice review. Kind of soaking in here because we're already familiar with all this part. Okay, the characteristics. All right, excitability, how responsive a neuron is to a stimulus. All right, the conductivity, how it's able to create and continue creating and making and sustaining an action potential. All right, secretion, we already saw that. Neurotransmitter release at the synaptic vesicle, at the synaptic knob into the synaptic cleft. Okay, extreme longevity. When you're born with them, you're pretty much going to keep them for life. Okay, and then they do not divide. Amitotic. Okay, except in extreme circumstances. I had to get into a discussion with a student about this several semesters ago, and so I just want to remind you, if you see it on a test, neurons don't divide. Okay, there's always exceptions to the rules for most things. I'll let you know when some of those are. All right, we covered this already, so I'm just going to kind of skip through this just to kind of quick review here. All right, the cell body, all right, our plasma membrane is going to encase the cytoplasm, in which we call that perikaryon. Okay, this is where graded potentials are going to be found. All right, and then we have our Nissl bodies there. Again, what are those? Ribosomes. Dendrites, unmyelinated, so on gross inspection, there'll be gray matter, or the short processes that come off the cell body. Their job, receive input, simple, and then transport, or excuse me, I keep saying transport, transmit that information to the cell body. That's it. That's all they have to do. Okay? Because at the cell body, we're hoping we're going to get that graded potential going on. All right, a couple things about the axon real quick here. All right, here you can see it's the longest process, the biggest process that comes off the cell body. And the axon is going to synapse, so now I don't have to say makes contact with. Now I can say synapse, because we all know what a synapse is. The axon is going to synapse with either another neuron, all right, or an effector gland, which is a muscle or a gland. Cool? Nice review, right? You guys know this. By the looks on your faces, you look bored. That's good. That means you understand, you got this information. All right, where the axon attaches onto the cell body, this is the axon hillock. That is where we initiate the action potential. All right, cytoplasm in the axon is called axioplasm. Plasma membrane in the axon is called the axolemma. All right, some axons will have branches that pop off of it. Those are called axon collaterals. When we get to the end of the axon, it splits off into these branches here. Okay, those are the telodendra. At the ends of the telodendra is where we have the synaptic knobs. Inside the synaptic knobs is where we store our neurotransmitter inside of the synaptic vesicles. Okay, once the action potential reaches the synaptic knob, it will then hopefully stimulate the release of the neurotransmitter into the synaptic cleft. But we need an action potential to do that. Okay? End goal is to get that action potential from one end of the neuron to the other end. All right, we already talked about the cytoskeleton. You've seen this picture here. Okay, now some new information. Okay, so let's talk about some of the different types of neurons here. Okay, so when I talk about structural classification, your brain should immediately think anatomy. Okay, and so we're going to now classify these neurons, all right, by the number of processes coming off of this structure here. What is that? What's that? Cell body. That's right. Okay, so now we're going to talk about how we classify physically, anatomically, all right, these neurons by the number of processes that come off the cell body. All right, so here's the one that we have been labeling this whole time. You didn't even know it. The multipolar neuron, okay, has many dendrites, one axon. All right, that's the one that looks like this. Here's the soma, here's the axon coming off, and then you got these dendrites coming off. That's the one that's in your atlas. Okay? So, it's the most common one, so that's the example that we like to use most frequently. The second type is the bipolar. Okay? And we're not talking about my best friend's mom. All right? Honest truth, though, I'm not making that up. All right? But here we have, all right, two processes coming off of the neuron, or excuse me, off the soma. So the soma's in the middle. You have one process coming off one way, the other process coming off the other way. One is a single dendrite. The other one is going to be the axon. Okay? We get to see this type, the bipolar neurons, quite a bit when we get to chapter 16 when we talk about vision. All right, when we talk about how visual images are actually created. So we'll wait until chapter 16 to get into more detail. But it looks kind of like that. Then we have unipolar neurons, okay? And in this type of situation, all right, you're going to have the process extending, all right, off of the cell body like this. One process, then it splits. Whee! One goes one way, the other goes the other way. Okay? So the peripheral process will split into many dendrites, and the central process is going to go onwards to the central nervous system. Okay? So it looks like this. So the cell body has one process coming off, and then it splits. All right. We see this type of neuron with a lot of sensory neurons, okay? And then finally, the anaxonic neuron, right, has the cell body, all right, and then a whole bunch of dendrites. Looks like a sunflower, okay? So it has no axons, as the name tells you, anaxonic. All right, questions about anything so far? Not too bad, right? And I apologize about my poor drawings, but my son would be upset with me. Okay, so that's the structural classification. All right, let's quickly get into a little bit of the physiological or the functional classification, okay? So when we talk about the functional classification, we are going to discuss the direction of the action potentials. Simple. Are the action potentials going from the periphery up to the central nervous system, or are they coming from the central nervous system out to the periphery? Well, we already know it does that. Sensory neurons bring information from the periphery up to the central nervous system, so that's input, okay? And then motor neurons are going to bring output or motor information from the central nervous system out to the periphery, out to the effector organs, okay? So again, we've seen this before. Sensory neurons, afferent neurons, are going to conduct what? Inputs from both the somatic. What's the somatic? Do you all remember what the somatic nervous system is when we're talking about sensory? Well, conscious perception. Yeah, conscious perception. You know, touch, taste, sight, sound, right? Temperature, all that fun stuff. Okay, so what we can consciously perceive. All right, and then we have visceral. That's the stuff that we can't consciously perceive. So it's going to take input from both the somatic and visceral receptors and send it to the central nervous system. All right, most are unipolar. Okay, that's that one picture that I just showed you. All right, here you have the cell body has one process, then you have the process splitting. All right, one has several dendrites coming off of it, and then the other one has the synaptic knobs. All right, and it's going to synapse onto something else. Okay, so most of those, all right, are going to be unipolar. A few bipolar, well, we're going to see that when we get to the eye. Okay. Then we have our motor neurons, the efferent. Think effect, EFF, effect. Okay, that's going to bring that output information from the central nervous system, all right, out to our effector organs through the somatic and visceral effectors. Somatic is the voluntary. Well, there's only one effector organ for the somatic. What is it? Skeletal muscles. Yep. Skeletal muscles. You're using all of them right now. And then our visceral has the remainder. Okay? Which is going to be cardiac muscle, smooth muscle, and glands. Okay? So then one of my favorite structures in the nervous system are the interneurons. All right, otherwise known as the association neurons. Just check this out. These first three words here are awesome. These neurons not only receive, but they process and integrate information from other neurons. They do this all on their lonesome. They're key. Well, check it out. Look how much. Look at this stat right here. They're found within the central nervous system, so the brain and spinal cord. But check it out. It makes up 99% of our neurons. That's huge. Okay? Huge, huge, huge. So they're going to actually allow the communication between our sensory division of our nervous system and the motor division of our nervous system. Hence interneurons. Okay? They're in between. These guys will be multipolar. That's the most common type of neuron. Okay, so they do a lot, and we get to see them a little bit more when we talk about. I don't really talk about them too much in chapter 12, but we get to some of the other chapters when we talk about reflexes. We'll talk about them. Okay, but for right now, there are going to be these neurons that are going to be found in between sensory and motor neurons. And they're going to help to facilitate, or in some cases, inhibit, all right, nerve signals. So that's what we're kind of seeing here in this picture. So we're talking about the functional classification. I'll start. Let me zoom out a little bit. All right. So here we have some skin receptors for touch, vibration, whatever. All right. Picks up a stimuli. Transmits that sensory information afferently with an A, all right, up through the sensory neuron. Okay, this is a unipolar cell. So you can see here the cell body, this structure here, we'll learn about it. It's called the dorsal root ganglia or posterior root ganglia. All right, it's a structure that's closely associated with the spinal cord. But it's a swelling here. What's a ganglion again? You remember? Ganglion? It's a cluster of cells. Cluster of cell bodies. Okay? Cluster of cell bodies. Okay? So we'll have a whole bunch of these cell bodies from these unipolar cells in the dorsal root or posterior root ganglion. So anyways, the sensory information travels up, goes into the spinal cord here, and then it will synapse, this sensory neuron will synapse onto this purple fella. That's the interneuron, okay? And then that interneuron will then synapse onto the motor neuron, okay? And then that motor neuron will then send its action potential down through the efferent branch, all right, out to, for this example, the skeletal muscle, for whatever type of, maybe it's a reflex to withdraw the arm back, okay? So here you're seeing, all right, how this all plays a role. Sensory information comes in, into the control center or central nervous system, and then the motor input or output goes out, okay? We'll go through it in much more detail. This is very, very kind of simplistic here. Okay, so one of the other cells, and we talked about it briefly when we did the tissue chapter for chapter 5. We got to the nervous tissue because that is the 4th tissue. Okay, remember? Epithelial tissue, connective tissue, muscle tissue, and nervous tissue. Okay, well, the neurons are one part, all right, of the nervous tissue. The other part are the glial cells, okay, or what we call neuroglial. They're non-excitable. What the heck does that mean? They're boring. You don't want to take them to a party. They're lame. You'll end up leaving them there. No, what it means is, all right, they're non-excitable. All right, they don't transmit action potentials. They don't have a membrane. They don't, excuse me, a membrane potential. Let me finish that thought. Okay? So the neurons are the ones that are going to transmit the messages, the action potentials. These guys don't do that. Their job is to support. All right? That's their job. Support. So they're going to help to support the neurons. The neurons are doing all, I shouldn't say doing all the work, but they're doing a lot of the important work. Not to say that the glial cells don't have an important job. So we're going to find them in our central nervous system and our peripheral nervous system. Okay? Now, even though these cells are smaller, there's way more of them than there are neurons. Okay? So if we're doing a volume measurement, all right, space occupying, all right, they will make up at least half. That's quite a bit, even though they're smaller. Okay, which means they got to make up for that being smaller by just having more number. Okay, to have that equal amount or that half volume. All right, now these guys, huge, they can divide. Okay, neurons can't. All right. So basically their job is almost like the police force of the nervous system. They protect the neurons, and then they're also going to help to provide sustenance for the neurons. All right? And in some cases, all right, they're actually provide a framework for the developing nervous system. All right? So you'll see in the beginning here, a lot of times the glial cells will occupy certain spaces, physical actual spaces, all right, in a developing fetus, child, and whatnot, and then eventually the neurons will then move into there and occupy those spaces where the glial, they're like a seat saver. All right, you ever go to the movies and then, hey, Bob, go in there and save us 3 seats, you know, right in the middle. So I'm going to go get my popcorn and my soda, okay, and my juju fruits. So that's what these guys do. They're a seat saver. They occupy the space and wait for the neurons to grow into and migrate into that area there, okay? All right. They also do play a role in the synapses. They can help with what we call neurotransmitter uptake. If you have too much neurotransmitter sitting out in the neurosynapse, all right, they can help to break down the neurotransmitter and they can help take it out of there. Because if it keeps sitting in the synapse, it can keep causing that nerve signal to occur when you don't want it to occur. All right, so we're going to talk about some of the different types of glial cells. All right, the first type, astrocytes. Okay, astro, think of a star. All right, these guys at the end, so they're star-shaped cell, and this is going to be a horrible picture, so kind of like just the different processes coming off. And at the ends of these processes, similar to like the synaptic knob, we call them the perivascular feet. All right. So this is the most numerous glial cell in the central nervous system. And unfortunately, you can actually get certain types of tumors, an astrocytoma, all right, that can occur in the central nervous system up in the brain from this type of cell here. But they play a huge role with this structure here. The triple B, the blood-brain barrier, okay? The blood-brain barrier is very important because it helps to keep out harmful substances from your nervous tissue, all right, in your brain. Because let's face it, we don't all make the best of choices. Sometimes we want to put some drugs into our body that shouldn't be there, or consume certain alcohol that some folks would say that shouldn't be there, all right? But point being is this blood-brain barrier helps to prevent the passage of a lot of harmful and possibly dangerous substances, okay? So it pretty much regulates what substances will have access to the brain, okay, which is good, okay? The brain is numero uno. Your body's job is to do whatever the brain needs done for it. So if we need to transmit, I keep saying transmit, if we need to transport more sugar or glucose to the brain, then the body's going to try to do that, okay? Not only do they help to form the blood-brain barrier, but they also will help with the tissue fluid, okay? Which is important because we were just talking about ion concentrations when we were speaking of sodium concentration and potassium concentration. So when we're dealing with certain ion concentrations, especially potassium, all right, these cells will help to regulate, all right, the amount of ions that are available to the central nervous system, mainly the brain, okay? All right, structural support, we just talked about the neuronal development, all right, and then one of the things I didn't get a chance to tell you about is that if a neuron dies, okay, these glial cells help to fill that space up, right, so nothing else can get in there. It's almost kind of like creates like a scar tissue, all right, in the event that you may have had a concussion and damaged some neural tissue there, and those neurons unfortunately died because they maybe didn't get enough blood supply there. They underwent ischemia, and so what will happen is these astrocytes will fill into that space and occupy that space. Kind of a neat phenomenon. All right, ependymal cells. Ependymal cells are these, think of them as a filtration cell. If you have like a Brita water filter, okay? Ependymal cells essentially filter out your blood to make this awesome stuff, cerebral spinal fluid, okay? And we're going to see these ependymal cells when we do the ventricles, which are spaces in the brain, all right, or cavities in the brain. So we'll see these ependymal cells in the cavities of the brain and in the spinal canal, or the central canal, excuse me, which is this tiny hole or a small canal that runs down the center of your spinal cord. And it's lined with ependymal cells, and they make cerebral spinal fluid. The microglia, these are like the white blood cells of the central nervous system, okay? So they're going to initiate, all right, the immune response. I compare them to the macrophage of the central nervous system. And what they do is they go around, and if there's any foreign debris or infectious agents, they'll phagocytize. What does that mean? That's the Pac-Man cell. Okay? So they'll go around and gobble it up. So if there's tissue damage, they'll go in and remove the debris. Like those guys that you see, I think it's the first of the month on the road there, picking up yard waste from people's properties. That's what these microglial cells will do. If somehow you get some bacteria or viruses that make it past, all right, certain protective mechanisms to the central nervous system, the microglial cells will attack, all right, and engulf those infectious agents there. Well, we talked about in the peripheral nervous system about the Schwann cells and the Neurolemma cells, and those are the cells that actually lay myelin down on the peripheral neurons. Well, in the central nervous system, these are the counterparts, the oligodendrocytes. All right, these guys will actually wrap up, all right, the axon for the neurons in the central nervous system. Here's the thing, though. In the peripheral nervous system, the Schwann cell will wrap itself around the actual axon. In the central nervous system, the oligodendrocytes shoot off these processes, and the process can wrap around an axon. So one oligodendrocyte can wrap around many neurons, but a Schwann cell can only wrap itself around one neuron, okay, or axon, I should say. So you'll see, all right, these extensions that come off of the oligodendrocytes, and they'll wrap around another axon. I'll show you a cool picture here. This next slide here has got a lot of cool pictures. Okay. Let me zoom in here. So here you can see, let me zoom out. All right, here's a bunch of the different cells here. Okay, so here's an axon. All right, here's an example of an oligodendrocyte. So here's one single cell, and it shoots off an extension, and then it wraps around this neuron over here. Meanwhile, this, all right, oligodendrocyte will also shoot an extension off and wrap around a different neuron. Okay? Meanwhile, over here, you can see this green cell. That is our astrocyte, and it's got these little processes, all right, the perivascular feet. Now, what I really like about it is it shows you these perivascular feet that are covering this capillary here. Capillaries, okay, are one cell layer thick vascular structures, okay? So it's usually simple squamous epithelium. And when we're talking about blood vessels and lymph vessels, we call it endothelium. So these squamous cells, all right, will have these perivascular feet attached to them. And so these perivascular feet can manipulate the position of these simple squamous cells. And so it can allow certain things to pass through, and at the same time, it can prevent other things from coming through. It can smush the cells together so really nothing can get through, or it can kind of pull them apart from each other so it creates little gaps and stuff can fly through. All right, kind of a neat thing here. What we're seeing here are the ependymal cells. Those are those cells that help to make, all right, the cerebral spinal fluid. So we'll find them, all right, in the lining of the ventricles in the brain here, okay? And what they do is they filter out the blood that's flowing through, and it filters out. Basically, what it does is it just takes out the plasma, which is the liquidy part of the blood. All right? It's similar. Cerebral spinal fluid is very similar to your blood plasma. Then here's your little microglial cell. This is the Pac-Man cell. So if there's any infectious agents, it'll kind of swarm in there and take things out. All right. Any questions about anything? No? Okay. So we talked about the central nervous system. I have to talk to you about the peripheral nervous system. Okay. So we talked a little bit earlier about the neurolemmacytes or the Schwann cells. Okay. Their job is to wrap peripheral neurons with myelin. Okay. What's the purpose of myelin? You need to know it. Speeds up the action potential. Okay, makes something that goes fast even faster. Okay, so we have satellite cells that are also going to be found in the periphery here. All right, and their job, all right, we're going to see them around the cell bodies. Where? In the ganglion. Okay. Remember, the cell bodies are the area of the cell that has all the stuff, All right, endoplasmic reticulum, Golgi apparati, all these organelles, especially the nucleus. Okay, so what the satellite cells will do is they're going to help facilitate the exchange of nutrients and wastes across the plasma membrane. And not only that, they're going to help to insulate the cell. Well, that's good, because remember, think of the neuron, all right, as a conductor. Taking physics, you know, you have conductors, and then you have, what's the other one? The resistors, okay? So conductors will facilitate the movement of an electrical signal, all right? The resistors will slow it down, okay? Well, these cells help to facilitate, all right, and promote the conduction of that action potential, okay? Well, at the same time, they're going to take care of the soma and all the structures inside the soma, all right, in regards to nourishment, and basically it's going to drop off the groceries and pick up the trash. Okay. Let me show you a quick picture here. All right, so this is where we're seeing, all right, in the peripheral nervous system. All right, in our peripheral nervous system for the neurolemmocyte or the Schwann cells, you can see, all right, Schwann cell wrapping itself around the axon. Individual myelinated coverings are going to be, all right, individual Schwann cells, and its job is to increase the speed of the action potential. Okay, then you can see here in our posterior root or dorsal root ganglion, here's the unipolar cell body here, and then you can see how we have all these satellite cells kind of surrounding it. And it does what we said, helps to insulate it, so as the action potential moves through, helps to facilitate the movement of the electrical signal, but also it helps to nourish the cell body and remove the waste from that area. I'm going to stop here for this because I want to do some more labeling for the muscles. And then we'll pick this up next class. All right, there's only like 8 more slides. Plus, I want to spend a little bit more time on myelination. I can see people's eyes starting to glaze over. I get it. You got tests on the mind here. So let's just do some simple labeling here. Okay. And then there's only like a couple muscles here, real quick. All right, so bust out your atlas. We're going to start labeling muscles of the head and neck. Does anyone have a page number for me? 21. Did I say that? 29. 29. Page 29. Okay, so our first, okay, the first muscle that we're looking at, all right, is the Frontalis muscle, which is part of a larger muscle group, all right, which is called Occipital frontalis. Okay, so you can call it the Frontalis muscle, or you can call it the frontal belly of the Occipital frontalis muscle. I think you all should do the second one. All right, promise me that you'll do that. Just joking. Okay, but again, next class, if I have time, I'll explain that a little bit more as to what the heck occipital frontalis is. But this muscle sits right on your forehead. This is the muscle that allows you to raise your eyebrow, or both eyebrows. You're familiar with Dwayne "The Rock" Johnson? He gave the people's eye. He had mastery over the contraction of the frontalis muscle. Okay, so it helps you to just raise your eyebrows up. Okay, so what it does is it goes from the front of the skull back down to the back portion of the skull. Okay, so you can see here we go from muscle tissue and then we go to something that doesn't look like muscle tissue. Okay, That's called an aponeurosis. And an aponeurosis is a flat tendon. It's essentially what it is. Okay? So we give that a name. Okay? We call that the Ganglia aponeurotica. Okay? Or you can call it the epicranial aponeurosis. But like it says here, it's going to link frontalis, or the frontal belly, all right, this guy up here, and we haven't seen it yet, to the occipitalis, or the occipital belly of occipital frontalis muscle. Okay, so you have one muscle up here, then you got this sheet of connective tissue, and then on the back, which we'll see in a second, we're going to see the other part of the muscle. But this connective tissue here is the Ganglia aponeurotica. Okay, right there. So the other part of that muscle, now this should be pointing to the red. Okay, that's the occipitalis muscle. All right, also known as the occipital belly of occipital frontalis muscle. That's this red area here. Okay. So now, well, before let me just show you something real. Well, I can't really see it very well. Well, I can see it here. All right. If you notice, when we look at these models here, all right, you can see that the right and left sides don't look the same, correct? So on our left side, and on the anatomical left side, you can see the skull, the bone. And on the right side, you can't. So I'm just going to let you know. The muscles look different from left to right. On the left side, that's the deep dissection, which means we've peeled a couple layers away. On the right side, basically what we've done is just peeled the skin off. Okay, so a lot of these models, excuse me, are going to have a superficial dissection side and a deep dissection side. If you see bone, you're deep. Okay, if you're not really seeing the bone, you're on the superficial portion. So I just want to explain to you why things look different. Okay, all right. So here we're looking at the left side. This muscle, temporalis, okay, is a deep muscle. It's also a muscle of what we call mastication. What is that? That is chewing. Okay, it helps to close the jaw. Okay, it is a muscle of mastication. So when you're chewing on your gummy bears, all right, tonight when you're studying, you are exercising temporalis. I love gummy bears, by the way, in case anyone was wondering. All right, temporalis, deep side. Now we're going to flip over to the other model here. This is the superficial side. So we peel the skin away, and we've got this other muscle here, smaller, similar in name. It is temporoparietalis. Okay? So those are two different muscles. Temporoparietalis. It's not a muscle of mastication, in case you were wondering. Okay. So I'm going to go through and describe, not tonight, all right, but in the next class, all right, certain appearances of muscles, why some look circular, why some look straight, all right? That's just based on the muscle fascicle arrangement, and we'll talk about that in the next class. But this next muscle here, okay, it looks circular in shape, all right? It surrounds the eye. So when you're squinting, when you step out and it's really bright somewhere and you start to squint, you are going to be exercising this muscle. So it's called orbicularis, like how a planet orbits the sun, circular. Orbicularis oculi, well, that's easy to know. That's the eye. Okay, oculi, orbicularis oculi, external muscle. It's the outer fibers here. Okay, so this is the orbicularis oculi external muscle. So on this next slide, now just to let you know, for your exam, this is when everyone pays attention to me, when I say for your exam, all right, instead of writing out the whole word muscle, all you have to do is write M period. That's it. Don't even bother writing out muscle. Yes, ma'am. Sure. You got it. Is that good? All right, so anytime you see orbicularis in the name of something, it's going to be circular for the most part, okay? So let's advance to here. Here's the other orbicularis muscle, okay? This is right around the eyelid here, closer to the eye. This is the orbicularis oculi internal muscle. So the external fibers are further out. Are going to be more towards the eye. Don't be scared. It's okay. It does that every once in a while. Pardon? It's right on. Yep, right there. If the arrow's pointing to the eyelid there, okay? Again, it's a circular shaped muscle. These fibers are just closer to the opening here. Okay, now, since we're talking about orbicularis shaped muscles, let's go down to the mouth. See, now you've got this muscle that surrounds your mouth in a circular type of fashion. So we call it orbicularis, O-R-I-S, like oral, but oris muscle. Let's say there's a circular muscle around the mouth. And this is the muscle, when you contract it, it causes you to pucker your lips. It purses your lips together. Okay? You ever tasted something sour, you're like, ooh. Or if you see somebody fall down a flight of stairs, you're like, oh, you know, when everyone does that. I think I married the wrong person, because when I fall down, she laughs at me. I'm not the type of person that laughs when people fall. When I worked in the restaurant, I was the first person if someone slipped in the kitchen. Are you okay? But no, my wife likes to laugh when I fall. Messed up. Okay, so staying here on the face, you're going to see a muscle that extends from the zygomatic bone. Remember, that's that cheekbone that's right over here. And it goes down towards the corner of the mouth. That's called the Zygomaticus Minor. And it's this guy right here. Okay, so if there's a minor, there's going to be a major, which I'm going to tell you a little rule here that I tell my classes. The minor is always above the major. We're going to see some other minor and major muscles later on. So Zygomaticus minor is here, so Zygomaticus major is going to be below it, down here. Okay? So the minor is always above the major. We'll see that again in the back when we're talking about the rhomboid muscles. It's the muscle that's in between your shoulder blades. Okay, so here's, again, and it's originating here on the zygomatic bone, and it's extending down towards the corner of the mouth. Okay, Zygomaticus major. So you're going to find out, and again, I haven't explained this to you, but I will. All right, we name muscles. There's many different ways that we name muscles. Some of the names are where they come from, or where they're going to, or what they're doing, all right, or what they look like. Okay, so this next muscle, we name it after what it does. It's called the depressor anguli oris. So here you can see it. It's below the mouth, but it's going to insert on the corner of the mouth, otherwise known as the angle of the mouth. Okay, so all these words tell you something about this muscle. Depressor is going to pull what? The corner of the mouth down. So this is the muscle that makes you frown. Okay? The Zygomaticus major and minor help to make you smile. Okay, but depressor anguli oris pulls the corners of your mouth down, so now you're frowning. Okay, right in the center here, you have two muscles coming towards the processes of your mandible. Okay, we call these muscles mentalis. Okay, two small muscles right there on your chin. Mentalis. And then we have another group of small muscles up on your nose, and that's nasalis. These muscles allow you to flare your nostrils. You can't, I don't know if I have it labeled here, but this muscle right here that goes along the side of your nose, this one here is called levator labii superioris aliquinasi. I just like to say that. It's not a muscle that you need to know for this class. It's a very small muscle, but it's got the longest name. Levator labii aliquinasi superioris. Basically, you don't need to know it for class. But it's this muscle that goes along the angle of your nose. But basically, that's an example of a muscle that it tells you what it does and where it's located. Levator labii, which is it elevates the lip. Okay. Aliquinasi means along the angle of the nose here. Superioris. That's all. Again, we'll go over some of these, how to name these muscles. I just like to throw that out there because people get freaked out. What? I have to know that muscle? Thank God, no. Okay. Another masseter muscle, or excuse me, another mastication muscle. This is the muscle that sits right here on the ramus of your jaw. Remember the mandible here, okay? It is actually the most powerful muscle of mastication. And if you don't get your tetanus shot and you step on a rusty nail and you don't do anything about it, that's the muscle that gives you lockjaw. Has anyone ever heard of that? Lockjaw from tetanus? You don't take care of it, right? So this is the muscle that you make very angry if you don't take care of the tetanus. Okay, that's the masseter muscle right here. Okay, next is risoris. That's this horizontal muscle. Again, when you are laughing, this muscle is stimulated, pulls your lips back. That's what risoris means, laughter. Okay, that's this horizontal muscle right here, in between the angle or the corner of your mouth and masseter muscle. All right, now what we're going to do is we're going to flip and look at the other side. This is all the superficial dissection. Now we're going to go to the deeper dissection. So I flip this over, okay, and this back in here is buccinator. So this muscle here is very well developed in folks that play wind instruments like a tuba, trumpet, trombone. But the purpose of this muscle is as you're chewing, okay, you'll probably start paying attention to it now after this class. But when you're chewing your food, this muscle contracts and it holds your cheeks close to your molars so the food doesn't fall out of your mouth. It keeps the food against your cheeks against your teeth as you're chewing, not so much where you're biting your cheek, but it helps to hold the food in so it's over your molars so you're crushing up the food. So that's buccinator. Okay, now we're going to go down into your neck. This next muscle is a muscle that's named after its origin and insertion points. This is Sternocleidomastoid Muscle. Okay? So the sternum And Cledo is stands for the clavicle. Okay, so it actually has two heads. Okay, it actually will insert onto your sternum and your clavicle, and then it travels all the way up and inserts on the mastoid process of your temporal bone right behind your ear. Okay, that's the sternocleidomastoid muscle. We'll talk about its actions and other stuff later on. Okay, staying on the deep dissection. Well, this is not on the deep dissection side, but this is actually superficial. Now we're going to flip over to the deep dissection side, and we're going to talk about the levator scapulae muscle. So if you see here at the external auditory meatus, you go just inferior to it. All right, the levator scapular muscle is going to attach just below that, and it's going to travel down on the back portion of the side of your neck. All right. So this muscle attaches onto the head and a couple areas of the vertebral spine there, and it'll attach onto your scapula. Hence, if you look at the term, levator scapula. It's going to elevate scapula. It does other things too, right, but that's what it was named for, elevation of the scapula. I'm going to show it to you from the posterior view. This is the lateral view. And now we can see it from the posterior view. This is on the deep dissection side. So it's this muscle that's peeking out right over here. And you can't see it, but the shoulder blade is down here. Excuse me. The scapula is down here. Okay, so I like to use the levator scapula muscle as a landmark. Okay, so if we go posterior to it, right behind the levator scapula, you got this muscle here, the splenius capitis muscle. Capitis is referring to the skull. So it attaches onto part of the mastoid process and part of the occipital bone back here. All right, that's splenius capitis. Here's the levator scapula muscle right here. So now if we look at the posterior view of our skull, there you can see the splenius capitis muscle. Its fibers run down and in. They move medial, down and in. Okay, then the most superficial muscle on the back of your neck and your upper back is this guy right here, the trapezius muscle. And this muscle is named after its shape. It looks like a diamond. So here's the head. It starts here at the base of your skull, goes out to your shoulders, and then it comes back in, and it goes all the way down and inserts onto T12. But it looks like a diamond or a four-sided geometrical figure. Well, what is that? A trapezoid. So we name this muscle trapezius. All right. So on this next slide here, on the posterior view, all right, this is only part of it. Okay, we're only showing you the part that goes out to the shoulder here. Okay, so it's here at the base of the skull and it travels out. That's the trapezius. So these fibers help you to shrug your shoulders. I don't know. When you bring your shoulders up, I have no idea. Okay, trapezius muscle. All right, now I'm going to introduce you to a family of muscles. I call them the Scalene Sisters because there's 3 of them. All right, this is on the deep dissection side. The most anterior one is called the anterior scalene. This group of muscles are going to be located in front of the levator scapular muscle. So then just behind the anterior scalene muscle, there's the middle scalene. And then finally, the muscle that's located behind the middle scalene, the last muscle that we're going to label tonight, is our posterior scalene muscle. So anterior, middle, and posterior scalenes, those are all in front or anterior to our levator scapula muscle right here. All right, folks, well, that's it for tonight. Okay, you can now rest your brains with this new information and continue on studying. I don't want to call it the old information, but for lecture and lab. Any questions from any of you folks at home? Okay. Well, since there aren't any, I wish you the best of luck. If you haven't taken your tests, good luck. And I will see you folks next week. Okay? But I know you'll be online on what's today? Tuesday, Thursday.